I'm Daniel Levine, and welcome to MitoCast. As researchers have taken a multiomics dive into the effects of travel in space on the bodies of astronauts, they've discovered that through the increased exposure to radiation and the effects of microgravity, it has damaging effects on mitochondria. In fact, the damage is similar to what happens to mitochondria as we age. As plans are being made for new trips to the moon, and there are discussions of a lunar space station and even trips to Mars, our ambitions for a future space age may rest on our success at figuring out ways to protect and rejuvenate mitochondria. And the development of such approaches, in turn, can have profound effects on our ability to treat mitochondrial diseases and address the effects of aging. We spoke to NASA principal investigators Chris Mason and Afshin Beheshti about what's been learned about the effects of space travel on mitochondria, why it represents a critical barrier for the future of space travel, and why solving the problem may give us ways of extending healthy years of life on Earth. Afshin, Chris, thanks for joining us.
Yeah. Thanks for having us. Yeah.
It's a pleasure to be here. Thanks.
You're both principal investigators with NASA. There are many authors on your papers, and others are publishing on the biological data being collected and generated by NASA. Can you perhaps start by giving us a sense of the scope of the research being done, the range of researchers involved, and what your roles in the research are? What makes your work unique?
Sure. So I'll kick it off. But Afshin and I have both worked on many projects over the years, and with Dr. Beheshti's work and many collaborators, we really stand here really on behalf of really hundreds of researchers across the world for some of these projects. But in a nutshell, the main goal is to better understand what happens to the human body and other organisms' bodies when they go into space. This includes microbes, plants, animals. Particularly focused in my laboratories a lot on human spaceflight. But we take a really broad multi-kingdom view of the biology. So whether it's human and microbial interactions, or how things change in your gut, for example, or genetics and epigenetics. So we take a broad view to look at the cellular, molecular, and full physiological changes that happen to the body to help us prepare for longer missions that are coming. And eventually start to also look at even countermeasures and precision medicine, which I think Afshin would talk about as well.
Yeah, sure. I mean, as Chris said, we work with many people. I mean, we do we embrace the whole open science and open collaborative network, global, because space is obviously can't be the space biomedicine world, the space biology world can't be researched by one person. Obviously, that has to be done as a global effort since it's so expensive and so many things are involved with doing it. So yeah, with my work specifically, embracing that whole open science area. We've worked with Chris, Dr. Mason. We've done many different projects together, not just covering, let's say, mitochondria, which we'll talk about later, but many aspects of the systemic impact of the body, since space is an accelerated model for aging and lots of diseases. So me specifically, I work on mitochondria, and that one aspect of unique things that I'm trying to figure out is the magic pill that you see in sci-fi movies. They want to take, they're like, oh, cured from all damage done in space. So maybe we'll make a cocktail, which I work with Chris closely with many of this stuff, obviously. So, and as we'll get into, we'll give some hints that potentially it's mitochondrial magic cure that we could take a magic pill to do that. It could be other things too, but that's the kind of the ultimate goal that I would push for, to say, while we understand the science of space on your body, the space environment to your body, really we need to figure out that how to make it safer, average people like me with a gut to go in space rather than the 0.001% fit people that are currently going right now, which I know is good that they're going now and I'm not going.
Well, Chris, you and I have spoken about your book, The Next 500 Years, Engineering Life to Reach New Worlds. Before we talk about what's been learned from studying mitochondria in space, it might be worth taking a step back and having you begin with the greater vision for the intensive study of the effects of space on astronauts and really why the survival of man may depend on our ability to go beyond Earth.
Happy to, yeah, and thanks again for the book plug. And it is out now wherever books are sold. You can get it. So it's basically a collection of goals, dreams, and aspirations of what I think humanity can do and also should do in the sense that we're the only species, as far as we know, in the whole universe that's aware of extinction and the frailty of life. It particularly stark in the vacuum of space and the stressors on the body and the radiation. But because life, I think about this almost every day, is that eventually the Earth will be engulfed by the sun. And so if we're still here, even if we have perfect global harmony and world peace and there's no war and everyone has enough to eat, and we will all still be burned by the sun. So I always take through the lens of a billion years, all ethical problems become quite clear. And I always think about this in terms of how do we start to look at the first forays into space, thinking about going to the moon or Mars, not just because we are curious, but because actually eventually we literally have to go that way if we want to survive in the long term. But in the short term, a lot of it is actually a forcing function for precision medicine here on Earth. So if you have to use very limited power, limited size, and ease of use, that's portable medicine. That's precision medicine in austere environments, for example. So, the book is about the why and the how. So eventually we go to the outer planets. We eventually go towards a new solar system and plan to live there. And also the ways you could even modify gene expression or genetics to enable organisms to live there, potentially someday even humans, because we have to wait and see. Maybe humans can survive just fine, but if we need help, we'll take any help we can get from any technology we can safely use.
Let's talk about mitochondria. We all know of mitochondria as the powerhouses of the cells and other critical functions, but to what extent are mitochondria limiting factors, both in terms of lifespans on Earth and then to your work for the potential for human life beyond the planet?
Yeah, maybe I'll take this one first. So from our work, that Chris and I have done is that we've shown, let's say this is back up for aging process. Everyone who ages, your mitochondrial declines. That's one of the hallmarks of aging. So that's, through the past few years, some scientists have defined 12 hallmarks of aging, and one of them is mitochondria. There's a bunch of, there's 11 more related with telomere shortening and so on. But I think I'm biased, and I think mitochondria is actually the hub of everything else downstream. But that's not what a common view, other than the fact if you become a mitochondriac, which hopefully this will convert everyone in the world to become mitochondriacs, meaning that mitochondria is the most important thing ever. But again, that's a biased view I have. So with aging, obviously declines. There's a lot of diseases that are attributed to mitochondrial dysfunction. For example, with cancer, there's the whole Warburg effect, where cancer hijacks, suppresses your mitochondrial energy production and replaces the glucose, glycolytic activity, which is bad for your body, but great for cancer to thrive and grow.
And even theft. The recent papers have shown that mitochondria can be stolen by cancer cells to kind of get more mitochondria, which is just kind of fascinating to think about.
Yeah, yes, exactly. So there's many diseases that you keep going down the list. Basically, a lot of the brain or CNS related issues, brain neural degeneration issues are related with mitochondrial dysfunction, meaning 20% of your mitochondria is found in your brain, and your brain's only 2% of your body mass. So imagine all that mitochondria packed in there, from Parkinson's to Alzheimer's. You just go down the list. A lot of that schizophrenia, even autism, has been shown to potentially be mitochondrial dysfunction related. So that's the clinical side. And now what we found with space is that from our paper, almost 4.5 years ago now, that we put out in Cell, that we saw, that was kind of the first paper in one emphasis to show that indeed mitochondrial is the central hub of dysregulation in space due to the whole environment in space of the radiation, microgravity combined. And then when you look at the parallels of what happens with diseases and aging, that's really there because, and now we're to have a follow-up paper that we're getting ready to answer the reviewers' questions. Hopefully it will end up in Cell again, but we'll see. But the idea there is that now we've done a deep dive looking at all the detailed mitochondrial pathways related with energy production to all the downstream metabolic factors and metabolism changes to how it impacts your immune system, and you go down to eventually to show that indeed you get that advanced aging phenotype due to the mitochondrial dysfunction, and all the diseases and the health risks associated with them is probably stemming from mitochondrial. Mm-hmm. Again, I could be very biased with my viewpoint here, but the data for me is pointing that way, and probably Chris might agree with me here.
Yeah, a lot of signs, all roads lead to Rome in the old days, sort of like all stressors lead to mitochondria and many diseases. And I think, in spaceflight, one thing that we really saw shocking was that mitochondrial DNA and mitochondrial RNA were both elevated when Scott Kelly spent a year in space, which was just really surprising. It was kind of just a really unexpected result. And we thought, well, maybe it's sporadic. It could just be this person. But now this Japanese space agency published a paper, Fumi, last year that showed a replication of this result, and we've seen it now for other crews. So this ejection is probably an interferogenic response, we think, or priming the interferon pathways, like the immune pathways of the body, is to prepare it almost for what it thinks is an infection or a stress. So really interesting to see this. Mitochondria is really the sensor as well as a mediator of the stress in the body. And we keep seeing it pop up, quite literally popping out out of cells and into the bloodstream.
Even for the shorter missions, like with Chris, all these commercial missions, we see the same thing happens. It doesn't matter if it's Scott Kelly for a year or Japanese agency's data that was there for 120 days on ISS. Even for the three-day short mission of Inspiration 4 that Chris was heading or in charge of, that a lot of the data showed the exact same thing. So it's like it doesn't matter. Space, the mitochondria has that same kind of stressor as Chris pointed out.
If the effects of space on mitochondria are similar to the effects of aging, what are we learning about mitochondrial stress on astronauts that have a potential to broaden our understanding of aging and ways of extending healthy life on Earth?
Well, I think a lot of that we can use it as a model for testing new drugs, new interventions, new therapeutics, because it is an accelerated aging model. We see a lot of the same stress markers show up. Epigenetic age, we've looked at this a little bit. That doesn't seem to accelerate too much, fortunately. But a lot of the other physiological phenotypes, for example, bone loss, muscle loss, cachexia, various phenotypes that look like aging, or sarcopenia. We've seen frailty markers. We've published also even kidney stones being more likely, or at least in the rat models. So there's a lot of interesting phenotypes, that things that you just get as you get older, and we can see them coming up at a higher risk for the crews as well as the animals that fly.
Yeah. So in terms of the effects of space on human biology, are the effects that we see on mitochondria the most significant, or are they just one aspect of our biology that's affected among many?
I think it's probably the most significant, but I think it's probably maybe the first line of impact to your body that causes, again, I'm biased to say this, but impact of it, because then people have shown also, and we've shown, and Chris has shown, and other people have shown, that all the immune function and inflammation is highly impacted. You see all these dysregulation, for example, of your innate immune functions being suppressed, and then inflammation goes up and cytokines get released, similar to like what happened in COVID, and SARS-CoV-2 infections. But I'm making the argument that mitochondrial is causing this downstream impact of these greater signals that you see later on. But the mitochondria has a great suppression of energetics happening across tissues long term too.
Mm-hmm, mm-hmm.
Yeah.
Some viewers may be familiar with the 44 article package of studies on spaceomics that was published in Nature. Can the two of you give a sense of the totality of what's been done to study the effects of space on our bodies and the bodies of other organisms, and how many subjects have been studied, what's been the duration, how granular or longitudinal look have researchers taken of the biology of astronauts in space?
Sure, because I'll jump in first. There are just about 700 people who've ever been to space, including some of the suborbital launches, which just get past that Kármán line of 100 kilometers. But for orbital flight, it's just below 700 of people who've been in space and orbited the planet for a while. And the mission with the Kelly twins, the NASA twin study, he was up there for 340 days, but there has been a Russian cosmonaut that was up there past 500 days in space. And so we know that there have been some longer missions, but not that many. Our knowledge of people in space for more than a few weeks is not that many. We have many 3 to 6 month missions, and by many I mean maybe really a couple hundred at this point, and that's about it. So it's really a few hundred examples of people that have been in space for months on end, 6 months, let's say, that we have data. We've published some of the papers with Dr. Beheshti that look at about 60 of those astronauts from some measures, like protein measures or cytokines, working with Dr. Scott Smith at NASA and other collaborators. But that, there's really probably, we tried to collect all of the data we could that's ever been publicly available, or that we published a lot with the Inspiration 4 crew, and get that into one atlas, the Space Omics and Medical Atlas sort of package of papers, mostly because this is something that's actually very common in cancer research and genetics research, where if you have a question and that you see a mutation, or you see a change, you see any kind of really molecular change, you want to know, well, is this common? Is this rare? Does this happen is this only in breast cancer? Is this something you see just as a function of age? Is this just stress? So generally, you need to have a large context and large number of samples before you can better understand your data. So we're trying to bring some of the tools of precision medicine and molecular biology to space medicine and also just general astrobiology, so the microbes in space. And we basically took every single data we could get that people had consented for and was possible to release into one atlas, and any mouse that had ever flown and rat. So you type in a human gene, you see all the changes that have ever been seen. You can also then look at the orthologous gene or the matching gene that's present in a mouse or a rat and have that plotted up as well. So we want to get every bit of data we possibly could into one spot. And this was made possible with GeneLab and the OSDR, the NASA databases, which really made it possible. So we wanted to make it so you can have a hypothesis and quickly look at it and access that data. And it was really also the crew, the Inspiration 4 crew, was very generous and said, we want to generate these data and get this out for people to use it. So a lot of the bravery from the crew and the generosity of the crew made this possible.
And one thing to point out too with the package is like these paper packages are kind of I enjoy doing them, and they're a lot of work for us to put in. But the good thing is, when you get the community to do that, like a lot of the papers that come out, even for me or Chris, are if you put them individually by themselves, sure, you read them, you're like, oh, this is a great paper. 6 months later, you might read another paper, but you've forgotten half the things that are in another paper, right? Because I remember these days, even the attention span of people are even shorter, right? But when you put the whole package together, like we have the table that Nature put nicely about all the systemic effect from the whole organism to the tissue to the cell to the molecular level, and then you see the pieces of the puzzle now as a whole together, as opposed to the bits and pieces of the pieces of the puzzle you might read every 2 or 3 months, which then you forget how these things are connected. And when you see the connection happen, now you're like, oh, now you see, let's say the kidney. There's like, for example, in this paper package from our collaborator Keith Sues in University College London, he did this massive paper about kidney, like 100 authors on there, the most comprehensive analysis that anyone's ever done on how space impacts your kidney. You read that, but then you say, how's that connected to, let's say, your cardiovascular, your brain? But I mean, you have the papers together, you're like, oh, there's like the whole mechanisms are actually connected, and the circulating factors might be involved this way. And I think that was the key, with everything Chris mentioned, how the data's there now, and then how all the papers come together. That's really neat to see, because now you see the systemic impact. And I think it really accelerates how people think about, how all these papers impact, potential countermeasure development or, understanding the basic science.
This is not like doing your typical clinical study. What are the particular challenges you face in studying mitochondrial health in astronauts? And do you do this in real time? Do you rely on biosamples and data being sent back to Earth?
Whenever we can, we do it as in real-time assays, in situ or like in-place assays right at that moment. And we've validated a sequencing protocol with what's called the nanopore sequencer. So that could be done in flight. And there's other assays we try to do in real time that we've just been validating, and flown a couple of times. But we would like to make it so you could do it in the space station or in the spacecraft. But often just getting blood draws, we get them either freshly drawn or you have to spin them down and freeze them. And so it is a challenge. I mean, there are logistical challenges. Samples have to be aliquoted, prepared, frozen, preserved, and then brought back and reallocated for investigators. So you have to run controls and then additional controls and ideally even more controls, because you really have to make sure that you're not just seeing a consequence of noise or some other just technical artifacts. Well,
yeah, and one of the things we rely on now is like a lot of mouse data, because obviously the astronauts are going to not give us a biopsy of their liver or kidney. Maybe in the future, Chris can vet. He's pretty convincing, so maybe Chris can convince him to do this in the future. But right now, we don't have that. That's the one limitation, No. As opposed to, let's say, in the clinic, if someone's getting a surgery for something, patient might consent. Oh yeah, if I'm taking a surgery in this organ, and it's safe, take a biopsy and give it to the scientific research. So we're a lot of the basic research is relied on the mice sent to space, and then now we could actually look at all the different organs, which is what we've been doing in our last paper and the most recent paper. That's you can find the preprint available. So then you translate everything Chris said to the human data of what's circulating, and that's, you could do that, and it's where you can piece it together nicely. But that was one of the, you talking about the limitations. That's one of the bigger limitations now for real time monitoring. But we hope, let's say, based on what we see in the mice, we see it translate to the humans. Then we could find the marker circulating due to mitochondrial dysfunction, circulating like the mitochondrial DNA, for example, is the low hanging fruit, or other factors related to what we see in the mice. And these papers were showing, okay, this is what's actually could be circulating to humans. And then Chris could go to the astronaut and say, oh, we could monitor your key mitochondrial factors for what's going to be, predicting your advanced aging or frailty that might occur due to the suppression that happens in your bioenergetics.
Well, are there things you think can be done to better understand the intricacies of mitochondria in zero gravity or under increased radiation?
Yeah, I mean, right now, for example,
Brookhaven, you talk about the Brookhaven.
yeah. I was going to mention that right now. Yeah, see, Chris and I think like. Yeah, so right now we do simulated experiments on Earth. So we go to, for example, NASA had set up a NASA Space Radiation Laboratory at Brookhaven National Lab. So Brookhaven National Lab is a high energy physics collider, where the physicists take particles, smash them together, and of course you get fundamentals of life and mass and what's there. And so there they actually produce the radiation types and ions you can get exposed to in space. So that's where in these heavy high energy physics, you can produce the iron particle, protons, and anything in between that the ions that that are in deep space the astronaut is exposed to. So then they set up a whole facility there where they took part of that beam and you could put irradiate mice, cells, anything you want, plants, electronics, and then you got that same kind of dosage that you might get in space. Well, in space, you get accumulated fractionated dose over time, a constant low radiation dose. You can't really do that on Earth, but we give like the total absorbed dose you might get, let's say for 30 days in space. You'll say that, or let's say a year round trip to Mars. How much dose that be? So we say, okay, well, zap the cells or the mice, with that, and then study them that way. So we do a lot of studies like that on Earth in those kind of environments. And then there's, of course, this microgravity, I put in quotes, simulators where for example, this thing's called clinostats, where basically random position machines that rotate. You put the cell in there, and it displaces the gravity to be microgravity in a sense. And then there's like things we do with mice experiments called hind limb unloading. You hang the mouse by the tail, which is silly, but kind of for a period of time, what it does is basically unloads the weight of the back leg, and then so this causes muscle degeneration, the fluid shifts, which you might see in the mice, and which happens in humans, when they're so there's some simulators. You could never truly simulate what's microgravity on Earth, but as humans, we gotta get close. So that's as close as we can get. And then we go to, okay, we know what happens. We potentially have maybe potential drugs or countermeasures, which we can get into later. That could potentially mitigate the mitochondrial damage. And then now we say, can we test it in astronauts? And then that's where Chris does his magic and gets his access to all the commercial missions to maybe test the potential countermeasures.
Yeah, that's what we're trying to do for some future missions, is basically use the accelerated aging environment of spaceflight and the stress of spaceflight, but use it as a rapid iteration platform for testing new therapeutics and countermeasures, including help for mitochondrial function. Chemferol is one of the molecules we've been looking at, but we're looking at any number of possible drugs and small molecule screens that could help survive in space, which could help on Earth.
Well, we appear to be on the cusp of a new age of space travel. There are new missions planned for the moon. There are discussions of a lunar space station, and there are visions for future missions to Mars. The notion of extraplanetary settlement, once the fodder for science fiction, appears now to be within reach. Gordon Friedman, the founder of MitoWorld, said to me the other day that no one's going to Mars until we figure out mitochondria. Is he right?
Yeah, absolutely. I would agree. So again, I'm biased, but yeah, no, I mean that's what we're showing right now. If I think we could predict based on our data we have, if an astronaut goes to Mars without any kind of countermeasure or precautions to take, their mitochondria will be so heavily suppressed and the bioenergetic damage that I don't know how they'll function. Like think of COVID, what happens with long COVID patients, which we're defining as a mitochondrial disease. Think of that multiplied by 10 or 20 or 100, by the time they get to Mars. I mean, the cognitive issues alone will be hard for them to focus based on the mitochondrial decline you get there. I would predict that. But, there might be hope because we're testing, as Chris pointed out, countermeasures to boost your mitochondrial signal, which has some very promising results that some of it's been in the preprint, but there's going to be a whole slew of papers coming out in the next year to show maybe promising results of that.
Let's talk about what it is in space that has such an ill effect on our bodies. There are two primary concerns. The first is the effect of radiation, and the other is microgravity, or the prolonged effects of weightlessness. Let's go through each of those. Broadly speaking, how significant is the exposure to radiation in space, and what effects does that have on our body over time?
I mean, the radiation, of course, you don't want too much radiation. I mean, some of it is unavoidable by just living in the universe. But if for example, the Inspiration 4 crew, got only about 4 millisieverts of radiation, and that was just over a few days. but when Scott Kelly was in space for years, 146 millisieverts, and that's the equivalent of getting a few chest X-rays every day, which again it doesn't kill you, but if you get that number 146, it's well, to put that in context. Once you get to about 600 or 700, that's about the expected lifetime exposure that you should get. So within just 1 year, he went to almost a quarter of what he should get for his whole life. So it is an accelerated risk, but in low Earth orbit, it's not too bad in the sense that we're still protected by the Van Allen belts. The Earth is still a really big shield for a lot of radiation that's coming from space, and so that is a helpful feature. But we're worried about the radiation, the fluid shifts, the isolation. You're also far from any help on Earth. The microbiome changes because you're in a really sealed environment, so that also changes how your immune system responds. We see a lot of immune perturbation actually in almost every mission we've looked at. So these are all factors we're considering that we know are going to need to solve or at least mitigate for the longer missions.
Yeah, and sorry to add to that too. Also, the exposure to the heavy ions in space. So something that Chris mentioned, the doses you'll get. So in space, you got something called in radiation biology, we got things called LET, which is a linear energy transfer. So the amount of energy the ion will transfer to your cells or tissues. So on Earth, everything that does your chest X-rays is gamma or X-rays. You got exposed to, and those are fast-moving particles that travel through your cells and tissue. And as they travel, they would transfer the energy scattered throughout the cells. But in space, you got high LET radiation, meaning you got these heavy ions from protons all the way up to iron particles of silicon. Just go through the periodic table, right? And then what happens is they're slower moving, but as they, let's say an iron particle is like a bowling ball. It moves slower, but then the energy just goes right into your cell, deposit all the energy into your cell as opposed to passing through. So now let's say an equivalent dose of a chest X-ray of high LET radiation, meaning the space radiation, and I'm just making up a number. Say 2 grays, the dose you might get for cancer therapy is a gamma radiation. If you got 2 gray of the high LET radiation, say of iron particles in the mixed beam up there, that's equivalent to like 10 gray or more of gamma, low LET. So it's almost lethal, getting that high dose. So the biology changes a lot. That equivalent dose that Chris mentioned is really important to understand that these heavy ion particles have a whole different biological interaction with your body than what people are used to getting that chest rate. So getting those 100 or what do you say about 10 chest X-rays, is actually a lot more damaging than you actually getting the 10 chest X-rays on Earth compared to the radiation equivalent in space. So that's the part where we're thinking that alone with what impact on the mitochondrial causes even a greater damage to the mitochondria as opposed to like the gamma that you might get exposed to on Earth, that equivalent dose you get.
How about microgravity? Or what would the long-term effects of a trip to Mars be?
Well, this is a big debate. I mean, we know it's not going to be pleasant on the body, but actually, I would even say the biggest challenge is not so much living in space, but it's when you have to land again, it's much harder on the body. We see a lot of the most significant spikes and changes the day of return to gravity. A lot of it is just the muscles and body realizing, the shock of suddenly having to use and build and rebuild a lot of the muscles and bone. So it is tough on the body. But I mean, one good thing about that is Mars is 38% gravity, so maybe it'll be 38%. As hard as it is landing back on Earth, we hope something. But it has other perchlorates in the soil and a different atmosphere, and you can't go outside. And there's things that might kill you there very easily in terms of the environment. So it is harder in other ways. But I think we want to plan to have these long missions with kind of all that in mind, I'd say.
So mitochondrial dysfunction underlies a lot of the ill effects that you've seen on the body. What exactly happens to the mitochondria in space?
So it's a whole cascade of events. So in our most recent paper, we've kind of really dissected that out with the help of one of the lead authors on there, Joe Garineri, who's like this mitochondrial expert. He's a young guy, who's helping us out on that. But what we see is a cascading event where the first impact of the mitochondria is your whole oxidative phosphorylation activity. So this is like your ATP energy production, whereas the different complexes, complex 1 through 5 is basically involved with all your energy production. So that's where we show, the longer you're in space was kind of intuitive, the more suppression of this happens. And then when when let's say mice or even astronauts come back from in the circulating factors, we see long-term suppression happen, even up to like from this Inspiration 4 data that Chris has provided. We see even up to 120 days as from the latest time point we had. It's the suppression of mitochondrial oxphos activity, so your energy production. And in mice tissue, we see the same. But then what happens cascade from this is that helps produce reactive oxygen species. So the key is like, reactive oxygen species can be produced from suppression of oxphos activity, but also radiation we know produces reactive oxygen species. So it's double impact, right? So that in turn creates hypoxia. So then hypoxic events in your body is bad. You don't want that to happen because that's going to create a whole slew of aspects to increase cancer risk, to cardiovascular risk, and you just go down the list. And then the cascade event after that, which is going to impact your immune system, where you get the inflammation that happens, and then also reduction or suppression of innate immune and adaptive immune activity that makes you more prone to infections now. So, for example, we see viral reactivation of not hepatitis, but herpes viruses happening in space, and other like potential viruses. Maybe people don't understand why would these viruses might become reactivated. Maybe it's because of the mitochondrial dysfunction suppressing your immune system, causing your infection, more prone to infection or reactivation of viruses. That has to be proven still, but that's a hypothesis maybe. And then eventually from that, it will cause cell death, apoptosis of cells, and then basically some mutations might evolve from mitochondrial DNA, to happen there. So this is in the current paper that's under review that we're showing this whole cascade event that happens downstream, which is very parallel to most things that happen in aging alone, but in an accelerated manner. Obviously with aging over 90 to 100, depending on the people live to 100 years now more. So depending that. But this is now over a short 30-day period. In mice, actually, we see this happen, in the space as opposed to over, your lifespan.
One of the other things the data has shown is that mitochondrial DNA is more susceptible to damage than our genome. Why is that?
It's a lack of DNA repair mechanisms don't exist as natively in mitochondria. So in our cells, we all get mutations every day, but almost all of them get repaired by enzymes and homologous recombination and other methods that basically look at the ends or broken ends of DNA and fix it and repair it. That's actually one great thing about DNA is because it has two strands. You always have a backup copy. If you look right across the pond of DNA, and so there's my other confirmation of my backup copy. So there's a really great feature in animal DNA and eukaryotic DNA, but we don't see that in mitochondria. And so they are subject more to the mutations. They have some protective mechanisms, but they can accumulate mutations faster. They also have something called heteroplasmy, where there's many copies of mitochondria per cell, and some more than others. But they have a whole dynamic range. We have half our DNA from our mom and half from our dad for their DNA with human DNA. But the mitochondrial DNA is all from our mother, and also it shifts depending on different cells. And as you just heard, like the brain has more mitochondria. So really depends on where you are in the body and what they're doing. There's different amounts of mutations and different numbers of mitochondria. So it's kind of like a hybrid ecosystem of DNA and genomes inside of our bodies. Much like plants have, their host DNA and chloroplast, very similar, where they have kind of a little ecosystem inside of them.
One area where mitochondria are rich are our eyes, which it turns out are particularly vulnerable to the ill effects of space. Can you explain neuroocular syndrome and what's understood about its causes?
Yeah, so yes, space-associated neuroocular syndrome, SANS for short, we call it. That class, well, originally it was thought supposed to be because of the whole microgravity environment, like you get the flattening of the edema, the fluid shifts that might happen. So like, that's going to cause where the issue that happens. The whole issue there is some astronauts get decline in vision over time. Some don't, and then some recover, and some are wearing glasses or get a thicker prescription when they come back. And some recover fully. So what makes some astronauts more prone to not have anything happen to in that mission time in their eyes versus some that do get the decline and some that have long lasting effects, some recover. Now, as you pointed out, there's a lot of not a lot, but there's some mitochondrial diseases that are associated with vision loss. Even macular degeneration has been linked to mitochondrial dysfunction, right, and suppression. So this is where we're showing in this paper also in mice, not in humans. We look at the eyes of the mice, and we see definitely that same whole mitochondrial cascading event happens in the eyes also. So in long-term effects that happens in the mice we looked at. What's interesting is that we're working on other factors related to clinical associated diseases, very similar to things like bone disease or and the mitochondrial disease related to vision loss. That whole cascade of events happen. Now, what really causes SANS, we're still trying to figure out. But I wouldn't be surprised in a year or two we say eureka, it's mitochondria that's
and it's different. Like the women seem to be less affected than men, which we don't quite know why yet. There's all sorts of interesting mysteries still about SANS, but more missions and more crew members kind of sharing their data will help us figure that out for sure.
Yeah. And I think truly, like if we maybe characterize the people before they go to space, their mitochondria, that might explain maybe the women have more resilience due to some mitochondrial trait, that's in them that men don't have.
Yeah.
But I think even the astronauts who might not be prone to the vision loss, I bet it's due to something mitochondrial resilience they have versus the ones who get the decline in the vision.
So at the top, when Chris was rattling off the different ill effects that was seen in astronauts from time and space, mentioned sarcopenia, which is the loss of muscle mass. Mm-hmm. I'm wondering if you can talk a little about the role that mitochondria play in this kind of space-induced frailty.
Yeah, so one of the works we're doing, one of the papers we're working on, and the paper we published last year was actually looking up frailty and the frailty index. So in a clinic, they as you grow older, they find you have your physical age, but not all people, in their internal clock might not be like 80 years old. Their body might be like a 70-year-old versus as an 80-year-old, you might be a 90-year-old. So that's their little frailty index in the clinic that a clinician might define that your true age is actually what it might be defined something differently than your actual age is due to the physical changes in your body. So, with sarcopenia, we looked at, indeed, in the literature, we know what happens in sarcopenia is that one of the factors is oxphos suppression in the muscle that happens with sarcopenia decline, and more happens. You see that bioenergetics in the muscle getting further damaged. So we see that same exact parallel happen in space. You could argue, granted, we haven't dissected out whether that's going to be microgravity or radiation have the greater impact. Through the simulated experiments we're doing on Earth, like we're looking at that now, I'm biased. And I think the radiation probably has a greater impact, causing the sarcopenia and mitochondrial dysfunction, versus the microgravity, just in the mitochondrial part of it. But again, that part still has to be really worked out. Which component of space causes the greater mitochondrial decline? But basically, that's what we're seeing, and probably Chris has something to add to it.
Yeah, and over what time frame? So I think we've seen there's a dose effect of spaceflight, where the longer you're in flight, the more pronounced some of these changes and markers seem to be in terms of their responsiveness. So we've seen the same idea of the right drug for the right patient at the right dose. It's kind of like in spaceflight, you want to be the right protections for the right astronaut, at the right dose, and some people might need more, some people might need less, but we're looking at all these markers now for the different crew members.
And how lasting are the effects of space on mitochondria? And does the recovery suggest anything about the ability to restore health to mitochondria from other sources of stress?
Yeah, I mean, right now, the longest time point we have from the humans, from Chris's data in Inspiration 4, we see 120 days later, circulating factors related to mitochondrial suppressed still. Now, luckily, it seems like, we looked at complexes 1 through 5, the whole oxphos related to energy production cycle of mitochondria, and it seems like what Chris had done was a single cell RNA-seq on this, so we know that different immune cells that look at. So it looks like some of the immune cells, like the T cells and B cells, might after 120 days they start coming back to normal levels. That's good, but then the monocytes were the ones that were really impacted with the mitochondrial suppression, that they still weren't recovering from the mitochondrial suppression we saw, which could be prone to infectious diseases and getting more prone to infections. So that's the key. And then the promising thing is from our countermeasure experiments we're doing, that Chris mentioned, Chemferl is one of the things we're looking at. It's from funding from this one Japanese company, Otsuka, which is trying to launch this nutrisupplement. Chemferl, which is a flavonoid, is found in a lot of things. You probably had a lot of Chemferl this morning if you had blueberries or you had spinach, kale, but they have a more purified version, and they modified it so it can be easier to get in your system. So we're showing from the experiments we're doing with Chris and other folks, our other collaborators, where did the simulated mouse experiments at Brookhaven, and then we also did it on these 3D organoids, basically tissues on a chip, through in Chemferl. And we saw that indeed, we're actually mitigating the damage caused by the space radiation impact simulated, and the microgravity. And it was really neat to see that indeed what's so far with the data we're showing, it's actually restoring a lot of the oxphos related activity, that's being suppressed. It's bringing it back down to normal levels. And for example, like our collaborator Rob Schwartz at Weill Cornell Medicine, he has this, these organoid models, like human, basically tissues in a dish. So you could create a heart, for example, that's like similar derived from human cells that are similar, but it's in a dish, like assimilated. So the heart actually and beats is similar, like how your heart would beat in your chest. So radiation, what it does is damage reduces the beats significantly. You give this chemferal, it's reduced it back to normal. And then from all the data we're analyzing now, we're saying, oh, we're actually restoring a lot of this oxphos activity and the metabolic factors. So it's really neat that, it's probably not just one mitochondrial supplement. It's probably going to be a cocktail of things we have to give. I'm calling it. We probably have to have a mitochondrial cocktail, and then give that to the astronauts to figure out what that is. And I'm sure Chris has plenty to add here.
Yes, well, and I think it's also organ weight. We can see like the damage to organs is less pronounced, or even just lessened overall. The stress markers are better off. So I think there's definitely room for this countermeasure or others to help make sure we can protect mitochondrial function.
Well, have you had to find ways to monitor mitochondria health, and has this given us any way to monitor the health of mitochondria on Earth?
Yeah, I mean, one of the ways we do it is there's something called the cell-free nucleic acid, where you sequence all the DNA and RNA that's in plasma when you look at your blood, and that gives us a really broad view of the number of changes that we see for many human genes, but also mitochondrial RNA. So that's one thing we've been looking at. But there's, we want to embed these metrics into a full health profile and full disease profile as well.
You talked about countermeasures and Camphorol, but some of the other stuff you were looking at were B vitamins. Is that right?
Yeah, I mean that's we weren't looking at that, but that was like people from like Brian Crucian from JC and Scott Smith. They've done a lot of studies where on the astronauts, like right now the only I think standard supplement they take is vitamin D, which is a mitochondrial booster, but it doesn't fully mitigate any damage, but it's good they take it still, But then there's a study done by Brian Crucian and Scott Smith where they did vitamin B, I think it was, I forget which version, and yeah, it'd be right, correct. And then they were showing, for example, they were looking at sand specifically, and it was able to restore some of the physiological changes that happen with sands by vitamin B3. Not it didn't fully recover everything, but it was really promising. That's good. Yeah, and that's yeah. I don't know, if Chris, you have ideas here? Yeah. Yeah, well, Chris is pondering this. I have an interesting one too, where my collaborator Cliff Calloway and Kate Flickinger from University of Pittsburgh, the hibernation. So the sci-fi movies where you see the person in hibernation. They wake up. Oh, okay, after 100-day traveling in space. There's something to that because from some of their research and some of the stuff that we've been starting to do with other kind of organisms that go in hibernation. And even this extremophile that our collaborator Jason Perdisky from Portland State University, it's called killifish. So killifish, for example, is found in Amazon riverbeds and African riverbeds where 9 months out of the year the riverbeds dry. There's like basically is mud piles. So then what the fish do, and then when they're fish, they're not extremophiles, but when the fish actually lay their eggs and then have their progeny survive, those embryos become extremophiles because they have like 3 diapause stages in the embryos. In the second diapause stage, what happens really simulates hibernation, where all these other people are studying it, where what he's been showing is that in that diapause 2 stage, the mitochondrial and metabolism basically shut down to zero. So the eggs are basically hibernated. So what we've done and what he's shown from like this the normal low LET gamma radiation, he could give him 50 gray to 100 gray, which would kill us if he get that. They're fine. Nothing happened to him. So they were like, wow, he had one day accidentally spelled 4% hydrogen peroxide, and I came back the next day and he's like, they're still alive. So they're very, they could survive in this anoxic environment. So they're very, in the hibernating state, they're very resistant to a lot of damage due to radiation. So we've actually now done some simulated experiments at Brookhaven at a very high dose, which is more than any human will get, after 10 years in space. They seemed fine. They were sent to space. They seemed fine. And the key is now why I think that my hypothesis is that we're in the hibernation state where this metabolism shut down zero. Basically, reactive oxygen species are not being produced anymore. And I think one of the key things we're showing from mitochondria is that reactive oxygen species and the suppression of oxphos causes this whole downstream events of things. Sure, you're still going to get DNA damage in the cells, but there's nothing to interfere now with the DNA repair mechanisms. So maybe in that hibernating state, why they can take 50 gray or we went up to 5 galactic cosmic ray, which is pretty detrimental to humans. Maybe that's why they could survive it, because now the mitochondria is like, well, we're fine. We just don't work at this point, because we're hibernating. I don't know, Chris, you have other ideas there, but I find that fascinating because I think it's really neat for another potential countermeasure that's truly exciting to look at.
Yeah, and changing their function, changing when they go to sleep or hibernate is they're all like levers we're trying to look at for mitochondrial function.
Well, you say it'd be an interesting countermeasure. Would that have implications on Earth?
Yeah, but radiation workers, it could be even for if you think of cancer patients doing radiotherapy in one spot, but you want to protect the other adjacent regions and keep them safe. So there's definitely some applications where we're using radiation or other risky sort of procedures, but want to protect the cells nearby or in general if you're in a high risk environment.
And as you think across the data, were there any surprising findings about the effects of space on mitochondria that have you thinking at all differently about mitochondria?
I think the neat thing we did recently was, does this only impact mice or humans, or is it other species? So we, on the NASA Gene Lab or the Open Science Data Repository that has all these omics data from all the multiple missions and species. So we didn't only look at mice and humans. We looked at C. elegans, worms, fruit flies. We even looked at plants. So plants have a whole different mitochondrial impact, but they still have mitochondria. And then the same thing is like we were showing that it's just, it's this is agnostic whether you're a plant, a worm, or a human. It has the mitochondria, doesn't care. Space doesn't care how it damages the mitochondria.
That's right.
So we're not too different from a plant or a worm, in space. When it comes to mitochondrial damage, that's basically what I found. I was like, oh, okay, neat.
Yeah, and my biggest surprise was just the fact that it was ejected from cells, that it's being found in plasma, because other cases where that's been observed is, for example, people who attempt to commit suicide and they don't succeed, fortunately, but then they go to the hospital and blood is drawn, their levels of mitochondrial DNA would spike up really high, which is interesting. Like the stress marker is normally been seen in really severe stressors, like an attempted take of your own life. But then when you look in the blood, you see the spike. So seeing this really, really stark sign of bodily stress, it was interesting to see where else we see that.
So we see the White House and NASA accelerating planning for manned missions. Is this going to happen without giant strides in mitochondrial improvements? And is this on anyone's radar other than mitochondrial biologists like you?
Yeah, I think we need to really target that. And if you don't address the mitochondrial cocktail for the potential, or the hibernation, or a combination of hibernation mitochondria, it's something related to mitochondrial research. Yeah, I don't think it's gonna, because I think a lot of times a lot of people might try to come up with a countermeasure that remedies what's below the actual source. So if you, let's say, come up with something that might help your immune system, you're still in the rescue of the mitochondria. So that's just a band-aid of what's truly happening to the actual source of damage that constantly is going to cause that immune dysfunction, for example.
So if NASA called on the two of you and said you needed to solve this problem in the next 5 years, what would you recommend?
Well, easy. We just need like $10 billion and a few missions. And no, well, actually, I joke, but not totally, because actually, the things we really want to do is do large scale screens of many molecules to look at the effects of radiation and microgravity on mitochondrial function. We want to fly missions that have, instead of 2 or 4 people, dozens of people up there, and like there's many space stations being built right now that might help us get larger sample sizes and do these kind of clinical trials and track people long term as they start going into space. So. And you could do that if you had billions of dollars. You could run these big studies on Earth and in space, and then do large countermeasures, do more screens, more synthesis testing and chemistry of other mitochondrial modifiers. And then, and if you do all that, maybe by 2035, we could head out to Mars.
And the key is, though, some people would be like, well, are you spending $10 billion to try to cure something that happens in for 4 or 8 people, fraction thing. The thing is, as I said earlier, this is an accelerator. This is an accelerator model for a lot of mitochondrial diseases, right? So any countermeasure we come to solve that problem can easily be applied back to what's happening with all the clinical mitochondrial diseases. Like the SANS issue can be applied back to all the vision loss issues that happen in space in the clinic. So, or just go down the list, all the cognitive issues that happen in cardiovascular. So that's where, like, let's say, the $10 billion investment that Chris said, that could actually really accelerate all the, mitochondrial diseases on Earth. And that's the key, to happen. Yeah.
And would you include aging as a mitochondrial disease in that regard?
Yes, of course. Yeah, we don't want to call aging a disease, but it's sort of it is. It's chronic disease, right?
It's certainly unwanted, let's say. Yeah, for most people, yeah, it's right. Or it looks like Tourette syndrome. Like if you have a weird tick, you can just have a weird tick. But if it bothers you, then it becomes a disease. And so I think most people are bothered by getting too old too fast.
Chris Mason, professor of physiology and biophysics at Weill Cornell Medicine, and Afshin Beheshti, director for the Center for Space Biomedicine at the University of Pittsburgh. Both are principal investigators with NASA. Chris, Afshin, thanks so much for your time today.
Thanks a lot.
Thank you. Pleasure.
To stay up on the latest news and research from the world of mitochondria, be sure to check in with MitoWorld regularly. For MitoCast, I'm Daniel Levine. Thanks for watching.